audible, you are visible, your presentation is visible, and everything is fine at your end.
Okay, okay. Then we will move ahead. Okay. So, good morning to everyone. Good morning.
Good morning. Participants are muted. We will unmute at the.
Good morning.
Okay, okay. So it's a pleasure to welcome you all in today's session of our summer school program on livelihood security through sustainable horticulture and allied enterprises. So I hope you all are doing well, and we are ready to dive into today's session. So first of all, let's introduce myself. My name is Dikshita Prajapati, working as an assistant professor in the Department of Fruit Science, SP College of Horticulture at Navsari Agriculture University, Navsari, Gujarat. So today we will diving into fascinating and most valuable topic of growth and survival of softwood grafting of mango, influenced by grafting month and growing conditions. So this is a valuable techniques in horticulture and plant propagations, and especially when it comes to improving plant varieties, then increasing yield and ensuring uniformity and orchards and the plantation. So whether all are already familiar with the basics of graftings, and today I will give you a deep understanding about the techniques, their advantages, and of course the step-by-step procedure involves. So as you all know that an inadequate supply of genuine propagating material is a major constraint in mango production, and mango mostly propagated by both sexual as well as asexual method, but the vegetative method is desirable because it produces true to type plant. So before that, I will give you a deep understanding about what is softwood grafting. So softwood grafting is the one kind of propagation techniques in which the desirable varieties or scion or new flushes or young shoots are grafted into compatible rootstock. So it is known as a softwood grafting. And for the rootstock, we can use 1.5 year old rootstock. So it is the most commercial techniques which is used by the farmers because it have a more advantages, and it is a traditional methods which have a more advantages, like it is quick, economical and effective, and of course grafts can be ready within a year. So softwood grafting gives an excellent response in initial success with least possibility of mortality, better and uniform orchard establishment. So the because of this, the experiment was designed to following objectives. The first one is to study the suitable period for grafting in mango, and the another one is to study the success and survival of grafting in mango. So coming to experimental details, the experiment was conducted at a regional horticulture research station at Navsari Agriculture University, Navsari. And here the treatment comprises two factors. One is the time of grafting and another one is the growing conditions. So time of grafting, which includes the softwood grafts, were prepared at different intervals starting from April to February. Means at interval from April, June, August, October, December and February, the grafts were prepared. And after preparing of grafts, the grafts were kept under two different conditions. That is growing condition. One in an open condition and another one is the polyhouse conditions. And so the treatment combinations was 12, and each treatment comprises 20 grafts, and the repetitions for the experiments was 3, and the total 720 grafts were prepared for at different intervals and kept at different growing conditions. So the morphological data was observed in different intervals starting from 30 to 120 days, and recorded accordingly, and I will present a result later on. First of all, I would like to share the process of preparation of softwood grafting. So you all are known that for a successful grafting, we need scion and stalk. Okay, so thus how you select the rootstock, it is most important. So first of all, you have to take the rootstock which have vigorous in growth and have there are 1.5 year old, particularly for softwood grafting. Okay, then you already know that the whatever rootstock we are using, that is healthy, then pest and disease free infestation. And of course, the rootstock which is used for grafting is about 40 to 50 centimeter tall and have a pencil thickness. You already know that. Okay. Now, coming to the cyan, so how to select the cyan? You can select the cyan of your desirable varieties which you want to prepare. So for that, you have to take the cyan from the mother tree first of all. Okay. So you have to choose that type of cyan which is identical to the mother tree. So you have to select it from the mother tree. Then after selecting the cyan, before grafting, you have to defoliate the cyan before 10 days for the proper union and uniformity in the grafts. Okay, so it is the most important procedure that you have to defoliate it. Okay, so after selecting cyan and rootstock, then after you have to prepare a cyan and rootstock as well. So how to prepare cyan? So first, I already told you that you have to defoliate it, and then after you have to prepare a cyan by cutting into two sides like a fork, which is already given in the photograph C. Okay, then after preparing the cyan, the rootstocks are prepared and the longitudinal cut was made in the rootstock. After preparing the cyan and stock, you have to prepare a graft. So how it prepared? So insertion of cyan into longitudinal rootstock cut, as it is shown in photograph E. And after inserting the cyan into rootstock, you have to wrap it. Okay, already shown in photographs that the union was stock and cyan union was wrapped with the plastic band. Okay, so after completing or after preparing the grafts was covered with the polythene bags to maintain the humidity. So in this figure, we can show that after 20 to 25 days, the cyan is emerged out. Means new flush, new leaves are emerged out from after the proper callus formation or after proper union. Okay, so it is visible to you. So after 20 to 25 days, the grafts will produce new flush, and at that time you should take some care that after preparing the grafts were covered with the polythene bag. So when after 10 to 15 days, you have to remove it. Okay, the polythene cover is remove it from the graft. If we are not going to remove it, then it gave the adverse effect to the graft. The graft will not sprout and other problem is there. Okay, temperature is arising and all. So you should remove the polythene covers which is covered to the polythene softwood grafts. Then after, you can see here. Okay. Okay, here you can see that this is the polythene cover, plastic band which is used to proper joint of softwood, proper joint of rootstock and cyan. So after that, after emerging out, you should also remove proper union. You have to remove the plastic bands also for its proper growth and to prevent the restriction of growth. And one more important thing, this is the leaves. Okay, this is the leaves which are produced below the graft union. So you have to remove it periodically, otherwise the union energy or food material is converted to developing those kind of unnecessary leaves. So you have to remove it periodically. Okay. Now, this photograph shows the performance of softwood grafts of mango under polyhouse condition. Now, coming to experimental result. So this table shows the effect of grafting month and growing condition on day required to leaf emergence, success of graft at 30 days, and survival of mango graft at 120 days. So looking to the days required to leaf emergence, the minimum days taken to leaf emergence was observed in June month for individual effect. Okay, while for growing condition, it was observed when the prepared grafts were kept under polyhouse condition. So here the individual effects are best. Looking to the success of graft at 30 days and survival of mango graft at 120 days, for the individual effect, the maximum success and survival of graft was observed when the grafts are prepared in June month. Okay, you can see here. Okay, and kept under polyhouse condition. This will give good result. Okay, but looking to the combined effect or interaction effect, the grafts which is prepared during June month and polyhouse condition gave maximum success and survival of graft. Okay. This figure shows the effect of grafting month and growing condition on incremental length of mango graft at 30, 60, 90 and 120 days. So here this figure shows the interaction effect for incremental length of graft. So here you can see that maximum incremental length was observed when the grafts are prepared during June month and under polyhouse condition. Okay. So this table also shows the girth of graft at interval of 30, 60, 90 and 120 days. Here you can see that the interaction effect is found non-significant for all the period, but the individual effect is found significant. So here the maximum girth was observed at the graft prepared during June month for 30, 60, 90 and 120 days, and the maximum girth was also found when the prepared grafts were kept under polyhouse condition. You can easily see here. Okay. This figure shows the number of leaves of mango graft at 30, 60, 90 and 120 days, and the similar trend was also observed with regards to number of leaves, and here the June month is found better and the grafts were kept under polyhouse conditions give better result. So coming to the conclusion, so based on the foregoing discussion, it can be concluded that softwood grafting done in June month performed well in all parameters like success of graft at 30 days, survival of graft at 120 days, incremental length, girth, number of leaves per graft, and the August month was found at par. So looking to the polyhouse growing condition, the polyhouse condition performed better as compared to open condition in all the parameters. So looking to the interaction effect, when the plants are grafted during June month under polyhouse condition gave best result as compared to other combination. So thank you all as we conclude today's session. So I conclude the presentation. So anyone have query or question, so they can ask me. Hello, ma'am. Hello, ma'am. Hello, ma'am.
Madam, is there any result of growth promoters in
Yes,
graft?
you can. Yes. Like growth promoter, you already know that NA is also used for growth parameters. But here, I am not using the growth promoters, but growth regulator, of course, growth regulator have a good effect on grafts. And one more thing, we have also experiment on anti-transparent. Okay, have you heard the name anti-transparent? You are, I think you all are well known anti-transparent. So anti-transparent gave good result for successful graft because it
Anti
reduced the transpiration and other vegetative parameter also increases.
kaolin or any specific anti-transparent?
Yes, kaolin we were used and we found best anti-transparent kaolin.
Okay.
Yes,
Thank
sir.
you.
Okay, thank you, sir.
Madam, I have one question.
Yes.
Madam, in mango and sapota, in saddle graft, which type of stock are used? How many
Your
months?
voice is, hello, Dr. Prasul, your voice is not audible properly.
Madam, madam, in
Yes.
mango and sapota, if you
Huh?
do saddle graft, the how many month graft are used?
For saddle graft.
Yeah.
For saddle graft, we can use about 8 month or 1 year old rootstock. 1.5 year also it gives, because it's a one kind of Inash graft. Okay,
Uh,
so 1.5 year old rootstock we can use.
okay, okay, madam.
Thank you. Any question from participant? Hello. Hello, ma'am. Okay, any other question from participant? So, anyone want to ask question? Then it's okay. Otherwise, we will conclude the session.
Thank you, ma'am.
Okay. So I will would like to express my gratitude to and thanks to organizer who giving me opportunity to be a part of this summer school program. I would also like to express my thanks to coordinators, faculty members, and of course participants for your active engagement and thoughtful questions. Thank you all.
Any announcement hereafter?
Thank you, everyone. The session is concluded.
Okay.
Ma'am, in 15